This is the final lecture on the series on how cells harvest energy. This is part 3 of 3, oxidation without oxygen, and the catabolism of other macromolecules. In the presence of oxygen, cells can use oxygen to produce a large amount of ATP, but even when no oxygen is present to accept electrons, some organisms can still anaerobically respire, using inorganic molecules as final electron acceptors for an electron transport chain. For example, many prokaryotes use sulfur, nitrate, carbon dioxide, or even inorganic metals as the final electron acceptor in the place of oxygen. Among the heterotrophs that practice anaerobic respiration are archaea, such as the thermophiles, and methanogens. Methanogens use carbon dioxide as an electron acceptor, reducing carbon dioxide to methane, or CH4. The hydrogens are derived from organic molecules produced by other organisms. Methanogens are found in diverse environments, including soil and the digestive system of ruminants like cows. Evidence of a second anaerobic respiratory process among primitive bacteria is seen in a group of rocks about 2.7 billion years ago, known as the Woman River Iron Formation. Organic material in these rocks is enriched for the light isotope of sulfur. Sulfur-32 relative to the heavier isotope sulfur-34. No known geochemical process produces such environment, but biological sulfur reduction does, in a process still carried out today by certain prokaryotes. In this sulfate respiration, the prokaryotes derive energy in the reduction of inorganic sulfates like SO4 to hydrogen sulfide, H2S. The hydrogen atoms are obtained from organic molecules other organisms produce. These prokaryotes are similar to methanogens, but also use SO4 in the oxidizing, that is, electron accepting agent in place of carbon dioxide. Early sulfate reducers set the stage for the evolution of photosynthesis. This image from your textbook shows a micrograph of the archaeospecies Thermoproteus tenax. This organism can use elemental sulfur as a final electron acceptor for anaerobic respiration. It is found in sulfur-containing hot springs, such as the Norris Geyser Basin in Yellowstone National Park, which is shown on the slide. I discussed fermentation briefly in the previous lecture. Fermentation is the use of organic molecules as the final electron acceptor in the absence of oxygen. Fermentation reduces organic molecules in order to regenerate NAD+. Two types of fermentation are ethanol fermentation, which occurs in yeast. Carbon dioxide, ethanol, and NAD+ are produced. There's also lactic acid fermentation, which occurs in animal cells, especially muscles. Electrons are transferred from NADH to pyruvate to produce lactic acid. Circulating blood removes excess lactate, the ionized form of lactic acid, from muscles. But when removal cannot keep up with the pace of production, the accumulating lactic acid interferes with muscle function and contributes to muscle fatigue. This is why warming up before performing vigorous exercise is recommended. Here this is shown in diagram form. Again, yeast carry out the conversion of pyruvate to ethanol, while muscle cells convert pyruvate into lactate, which is less toxic than ethanol. In each case, the reduction of a metabolite of glucose has oxidized NADH back to NAD+ to allow glycolysis to continue under anaerobic or oxygen-lacking conditions. The previous lecture focused on the details of aerobic respiration of glucose, which organisms obtain from the digestion of carbohydrates or from photosynthesis. Organic molecules other than glucose, particularly proteins and fats, are also important sources of energy. So the next few slides will focus on the catabolism of protein and the catabolism of fats. This slide specifically focuses on the catabolism of protein. Proteins are first broken down into their individual amino acids. The nitrogen-containing side group, the amino group, is then removed from each amino acid in a process called deamination. The remainder of the amino acid is converted to a molecule that enters glycolysis or the Krebs cycle. For example, the amino acid alanine is converted to pyruvate. The amino acid aspartate is converted to oxaloacetate. So different amino acids may enter the process at different points. This image from your textbook shows the deamination of glutamate. So remember, after proteins are broken down into their amino acid building blocks, the amino groups are removed from the amino acids to form molecules that participate in glycolysis and the Krebs cycle. So for example, the amino acid glutamate becomes alpha ketoglutarate, a Krebs cycle intermediate, when it loses an amino group. This slide focuses on the catabolism of fats or lipids. Fats are broken down to fatty acids and glycerol. Fatty acids are converted to acetyl groups by beta oxidation. This is an oxygen-dependent process. Respiration of a 6-carbon fatty acid yields 20% more energy than a 6-carbon glucose. This diagram from your textbook shows beta oxidation of a fat. Through a series of reactions known as beta oxidation, the last 2 carbons in a fatty acid combine with the coenzyme A to form acetyl-CoA, which enters the Krebs cycle. The fatty acid, now 2 carbons shorter, enters the pathway again and keeps re-entering until all its carbons have been used to form acetyl-CoA molecules. Each round of beta oxidation uses 1 molecule of ATP and generates 1 molecule each of FADH2 and NADH. This is another one of my favorite diagrams from your textbook because it shows the degradation or breakdown of each macromolecule that you consume in your diet: nucleic acids, proteins, polysaccharides, and lipids and fats. All eukaryotes and many prokaryotes extract energy from organic molecules by oxidizing them. The first stage of this process, breaking down the macromolecules into their building blocks, yields very little energy. The second stage of oxidative or aerobic respiration extracts energy primarily in the form of high-energy electrons and produces water and carbon dioxide. Key intermediates in those pathways are also used for biosynthetic pathways, shown by reverse arrows.